Yeah, welcome back, everyone. So in session 2, we will have shift the talk to other use cases and analysis too. And in the first talk, we have the talk on the molecular surveillance tools for Plasmodium Vivax, which is given by Associate Professor Sarah Auburn, Menzies, from the Menzies School of Health Research. Thank you. Over to
Thank you, Brian, for the introduction. It's a real pleasure to be here today to speak with you about some molecular surveillance tools that we're developing within the vivaxGen network. And these are specifically tools that we've actually developed primarily to try and understand more about Vivax treatment failure. But as I'll show in a few of the slides, we believe we can also use them to address some other transmission-related use cases. So for the sort of storyline today, what I thought I would talk you through is, first of all, describing some Vivax surveillance gaps, and particularly ones where we feel that genetics could be helpful. And then discussing those tools that I mentioned around developing surveillance tools to understand more about treatment failure, including some laboratory assays that we've just recently completed development of, and then show you a little bit of pilot data from those laboratory tools from a clinical trial. And then in the second part of the talk, or the third part of the talk, just run through how we also feel that despite these tools having been made primarily for looking at treatment failure, how we think they can also be informative down the line on other transmission-related use cases for Vivax. So it's actually really nice to be able to talk with an audience who really understand the burden of Vivax. I think for this particular audience, you'll be very familiar with why we really cannot drop the ball in terms of our efforts to contain and eliminate Vivax malaria. You'll certainly be very aware that these case numbers that we get from WHO reports really don't give justice to the actual burden of Vivax malaria in regions outside of Africa, including, of course, the GMS. And certainly in the GMS, we'll be very familiar with these patterns we're seeing. We look at these WHO maps, seeing that the number of, or this sort of gap between all case malaria in red and falciparum cases in green, that's really widening as we reach elimination of falciparum because, of course, of this increasing predominance of Vivax cases. And I'm certainly hearing in this meeting and through other avenues of increasing reports of outbreaks of Vivax malaria, showing us how unstable this species can be. And of course, that's pretty concerning in terms of resurgence potential and, of course, the clinical impact for the communities that are affected. We know, of course, that a large part of the challenge with Vivax malaria is, of course, the dormant liver stage that really supports this parasite being able to, it enhances its transmission within and between communities. And as I'll describe in further slides, of course, it makes cure of this parasite particularly problematic. If we think about the blood stage treatment, as we'll all be very well aware, in many regions of the globe, we're still using chloroquine for treating Vivax malaria. But if we look at some wonderful tools like this Vivax surveyor map, we can see in regions across the globe these increasing reports of resistance to chloroquine. But even this map is quite difficult for us to truly appreciate and understand, because we know, of course, a lot of these reports, or some of these reports may well be confounded to some extent by reactivated liver stages. Unfortunately, in contrast to the situation with falciparum, we don't have good or effective markers for chloroquine resistance, or indeed many of the drugs that are widely used for Vivax. So about a decade ago, maybe a bit longer now, several of us within the APMEN Vivax working group came together to develop what we call vivaxGen as a sort of framework for propelling the development of molecular surveillance tools for P-Vivax, because we saw all of these gaps in the availability of surveillance tools there. So very similar to what's been done in GenRe-Mekong, but with a focus, of course, on Vivax malaria. And we took a leaf from GenRe-Mekong because, as we've heard this morning, GenRe-Mekong, and as Estee said, is really flying the flag in terms of what we, how we approach MMS. And we've seen this lovely plot here from Oliver, highlighting some wonderful use cases and really understanding that mapping between what program officers do and how we can try and support those activities from the research side of things. So we're following a very similar pattern in terms of what sort of use cases we could generate for Vivax. And for today's talk, what I really wanted to focus on, as I mentioned before, is really some tools we've been developing to try and address this particular use case, which is understanding better treatment failures, or more specifically, trying to get a better characterization of recurrent infections. And I've got an open star here because even though we don't have effective molecular markers, these two are somewhat connected, because if we can't actually define recurrent infection very well, so discriminate recrudescence from relapse and reinfection, then it's going to be really difficult for us to evaluate any new candidate markers or detect new resistance markers. And then, as I say, I'll just go through a few of how we believe these same tools can also inform on some of these other use cases. So first, if we just focus on the treatment side of things. And again, this audience will be very familiar with the concept, of course, when we're thinking about Vivax. We don't just think about the blood stage. We also need to think about the liver stage treatment to achieve radical cure. As I mentioned earlier, though, and I think you'll all be very familiar with this concept, that we just don't have any good effective molecular markers for any of these widely used drugs. So, of course, a lot of what we know at the moment about Vivax treatment efficacy or failure is really from clinical trials and cohorts. But even with the very best clinical trials, the best designed, most carefully designed clinical trials and cohorts, of course, we face this challenge that when in some cases, when we have a recurrence, we don't know whether that's a true blood stage failure, reinfection, or a reactivated liver stage. And we can't do that simple sort of 3-marker MSP1, MSP2 blurb type genotyping that's done for falciparum to basically classify our de novo recurrent infections the same or different, because of course for Vivax, we know that these reactivated liver stages can fit in either of these two classes. So what we've been doing is trying to reframe that question, and rather than simply ask a binary same or different, rather ask, between our de novo and our recurrent infection, how related are they? And I've got a family tree here. This is a human genetics one, but it's a very similar concept for the parasite, just to emphasize that we're thinking about that in a familial term. So does it look like our de novo and our recurrent infections share parents? And you might be wondering, why do we care about that? And the reason is we make this assumption that parasites that come from the same mosquito inoculum, because we have that obligate recombination in the mosquito midgut, even if they're different, so the same as me relative to my sister, we're not going to be genetically identical. Even if they're different, they should share large regions of the genome identical by descent. So if one of these parasites comes out at day 0 and the other comes out at recurrence, if we did the classic MSP1-2 type classification, we might say, well, they're different, it's probably a reinfection. But if we look at identity by descent, which we can use to measure relatedness, we'll see they're different, but actually they look quite related. And we think these come from, they share parents and they come from one inoculum. And so we can now actually rule out reinfection as a likely cause. We'll be thinking, I'm sure, well, what happens when we have identical parasites? Still difficult to tease apart recrudescence and relapse. And in these situations, though, there are some clever mathematical models developed by Amy Taylor and James Watson and others that take that time to event information to try and help to disentangle those. So what sort of assays do we need to capture that relationship between our day 0 and recurrent infections? We were quite fortunate that actually when we started this work, Amy Taylor had just published a study. I'm not going to go into detail on that. It's a modeling study that basically told us we need about 100 multi-allelic markers to capture that relationship. So what we did as a collaboration with Sasha Siegel and others from the Sanger Institute was to pull out what we call microhaplotypes. I think I'd heard that term used earlier in the day. In case it helps to show a schematic, basically what we mean is little windows of the genome where we have 3 or more SNPs. And you can see that that gives us all sorts of combinations. So we just capture a lot of genetic information. So we searched the genome to try and identify 100 of these microhaplotype windows. And we're also very conscious to make sure there were microhaplotypes that would work in different regions of the globe. And I haven't gone into detail about how we selected them, but if anyone's keen, you can find out in our preprint. And in case that was still a little bit too technical, I appreciate it's a technical concept. One way you might want to think about is that it's almost like we selected 100 NSP1 or 2 or GLP type markers. That's how much information we get, having about 100 of these. But of course, we're not going to do the genotyping by PCR and gels because that would take us forever. So the assays we put together are amplicon sequencing assays, very similar to what's used for GenRe-Mekong. And for this, the beauty of this is really that we can amplify all of our markers, the 100 markers for a single patient in one single tube. And then we put these little tags on that tell us this is from patient 1. And we can do that for several hundred patients and pull all of this together and sequence them in one go. So I was going to say actually that the beauty of this is that we can genotype many samples, many markers in parallel using very little DNA. And so that's great for high throughput. But actually this morning we've also talked about, what happens actually when you do want to go to a lower throughput. So I might mention that, we should also consider that there might be other platforms that we could use to scale down. So for our Vivax panel for amplicon sequencing, we've managed to get 93 microhaplotypes to sit in one of those tubes in one single reaction, along with one Plasmodium species marker to confirm is it definitely Vivax? Is there anything else in there? And then a few drug candidates. But I put these in gray because I'm not really sure that these are actually that informative for the control program. Obviously, happy to hear your thoughts on that. And then for the sequencing, we've been using the MiSeq platform. We chose this because it's actually the platform that many of our partners in endemic countries seem to have ready access to. But of course, one could use higher throughput platforms as needed. And then we use about 20 nanograms of DNA, and that's the composition of both human and parasite DNA. So we then did some sensitivity analyses to see how low can we go, because we know Vivax parasites often very low density. So what we did is take a few samples, such as this one here, KV3. This started at 7,000 parasites per microliter, and we went all the way down to just under 1 parasite per microliter. Sequence these along with several hundred other samples on the MiSeq. You can see to interpret this plot, if one said, for example, I'm happy with just 25, having at least 25 reads for each marker, you can see you'd capture most of our markers if you had 70 parasites per microliter. If you can get away with less, with just 5 reads per marker, you can see that now you still get a lot of data, even with just 7 parasites per microliter. And we see similar patterns with other serial dilutions. So in a nutshell, what that tells us is that we should be able to use these assays even on low density infections. We've put together a data processing framework, and I didn't want to go into too much detail here. Just to flag, these are the wonderful people who are helping with this data analysis process, and particularly wanted to highlight Dan Menzies' team from the Broad Institute. So we're actually basing this platform on a pipeline that Dan previously developed. You can find out more in his paper here that uses this DADA2 approach for the variant calling. But in this side of the work, a lot of it still, I'm very conscious, it's still very much in that research world. So we've made good progress on that research side of things. But of course, we're very conscious that we now need to take those research outputs and translate them into more actionable feedback for the NMCPs. So I'm really thrilled that we're starting to work with NIAN, and we're hoping we're going to be able to generate data from the GMS on the Vivax isolates there, and then also, of course, working closely with Oliver to understand how can we generate these useful epidemiological type maps for that data, but that's still in process. So in the interim, if I can show you a little bit of pilot data, we're still not in those wonderful actionable output formats, I'm afraid, but just to give you a flavor of what kind of information we could get. So this is actually a study that was published a few years ago. Lawrence actually was very much involved in this study. A randomized controlled trial conducted at 2 sites in Ethiopia, comparing chloroquine to AL and then those 2 blood stage treatments with primaquine regime. So just under 400 patients were followed for a year. And you can see here in this lovely survival curve, as you'd expect, when you add primaquine, much lower risk of recurrence. But what we really wanted to know was which of those recurrents are actually relapses, what might be reinfections, and so on. So from about 720 patients or positive cases, we lost some where we had no DNA, but managed to get 460 odd genotypes, which equated to about 130 patient pairs plus. I refer to pairs plus because, of course, some patients over that 1 year follow-up had multiple recurrences. And if we just as a start look at that day naught and the first recurrence, we can start to get a bit of information. Here we're looking at the relatedness between those pairs. And we can see, there's a few what might be reinfections down here because they're very unrelated and similar frequencies between the different treatments. And then these ones here, highly related but not identical. So probably a few relapses, mostly the relapses here. And you can see fewer of these in the PQ arms than in the non-PQ arms. And then right at the top, we've got these near identical infections. And that, of course, is where the modeling is going to be really key to tease apart recrudescence from relapse. And then we can start to get a bit of a taster for multiple recurrences as well. This is a cluster plot showing just from one of the 2 study sites, because they're very busy, cases as circles, and they're color coordinated by the patient. And when they're connected, that means they're highly related. So it might be a recrudescence, might be a relapse. And we have one example here of a patient who had multiple recurrences, and you can see they're all connected. Another patient here, just to give you an idea. And then another patient here who's had multiple recurrences, but you can see some of them are probably reinfections. So that's all nice and pretty, but it's still very much in that research domain. And we're now working more closely with Amy Taylor and James Watson and others to try and translate that into more actionable feedback. So lastly, as I mentioned, although we didn't specifically design these tools to look at transmission use cases, just because we capture so much diversity, we can start to address some of those. And we've seen this plot here earlier. As I say, because we can distinguish infections very well, we've got enough diversity. One thing we can capture is when a particular strain is expanding very quickly in a population. And we actually showed that before. So we did a study a few years ago where genomic data showed a clonal expansion in Sabah in Malaysia, just before Sabah eliminated Vivax malaria. And you can see here, if we type those same samples with our microhaplotypes, we can capture that outbreak. And even a few samples where we couldn't get genomic data, but with microhaplotypes, we can see they also belong to the outbreak. So that's really promising for early detection of outbreaks, and maybe also for determining when you have an outbreak, is there one or more source of infection? We also see that the markers have good geographic discrimination, so might be useful for looking at imported cases. And I'm keen to hear more from Tessa's talk about other things we might do in this space. I'll just try and go through these quickly. Sorry, I'm conscious of time. But one of the things we're doing is trying to see if we can adapt our online classifier tools so that we could determine when we have imported cases using those tools. And then because we can capture relatedness very well, we should also be able to characterize gene flow fairly well. We haven't looked at this in a lot of detail. Just to show you a little bit of data here, though, we should be able to, as I say, even within a country in a particular area, get an idea of the connectivity between communities, which might be useful for assessing risk of resistance spread and defining spatial boundaries for interventions. And then lastly, again, because we have so many markers, we can capture diversity within an infection very well. And as I'll show, that means we might be able to look at transmission intensity, This is just one polyclonal sample to give you an idea across the 93 microhaplotypes. You can see a few markers with evidence of multiple strains. So we would expect this type of infection probably comes from a sort of superinfection type event. And if we can get population level information, we can get an idea of how often this is happening and therefore transmission intensity. But really the utility of these types of tools comes in when you have quite high resolution data. So in summary, I've shown you that we have developed these microhaplotype tools and hopefully been able to show you that they have multiple use cases, not just for transmission, but also for treatment rather, but also for transmission. We have SOPs available and more than happy to share those with you. So if you're keen, please come and contact me. We're very excited that we've already started implementing them in Indonesia and really thrilled to be able to start working with Nian's team and other partners as well, bringing them in to the GenRe-Mekong sites. As I showed, we're also working towards the data analysis framework, but the key point here is that in the next year, what we really need to focus on is translating that to actionable feedback. And that's where your help is really going to be greatly appreciated to make sure we do that well. Many people to thank, and of course the funders, particularly the Gates Foundation, for wonderful support from ESTE. Thank you.
Thank you very much, Sarah, for quite a tour de force there. Very, very interesting. Can we have questions? Oh, Dr. Sarah.
I still not capture well, so that's why I ask. You want to tell us that the relationship of the same mosquito infect people or what?
Yes, sorry, that was probably a little bit complicated. So what we want to know is we want to know if you're looking at a clinical trial or cohort or therapeutic efficacy survey, and you have a sample from day 0 and at recurrent time point, we want to know, does it look like, they came from a single mosquito bite, or does it look like they came from 2 mosquito bites? Because that might help us to understand, is it really coming from the liver, the reactivated liver stage, or is it a whole new infection?
So when they are come the same mosquito and link together here, and you say that it is most likely it's a relapse.
Yes, yeah. Certainly if it looks like they come from the same mosquito, we'd say it's more likely a relapse than a reinfection. Recrudescence, blood stage failure, like if you had chloroquine resistance, that's still tricky. And we have to do the modeling and look at time to event. So we'd be anticipating very early time to event.
My question is, even the adherent or drug different. So different people will be the adherent with the drug will be different. Some may be complete dose, some not complete dose. So
adherence. So
yeah.
So these particular tools won't tell us whether the patients adhered, or I guess they might indirectly tell us whether you adhered, because if we see evidence you've got a relapse, that might tell us, yes, you perhaps didn't adhere to the...
You understand my question? My question is most likely the adherent of drug low, most likely you get the relapse, more relapse.
Yes, sorry, yes, that's true, yes, especially for... You're probably thinking of chloroquine?
Also, maybe because you want to see the same original infected by mosquito to see link together. But actually, some patients, the adherence is low. Some patients, the same infected mosquito, some patients receive good adherence. So how can they link together?
No, I understood that question.
So I think, and I don't know if it's the same thing or if it's sort of related. I mean, so if you're doing a sort of very controlled clinical study, then of course you will have, you may have parasite on the initial infection, and then you will be able to connect it to the same patient later on. Then you can tell, is it a relapse or not? what sort of what I'm trying to imagine is like if instead you are doing surveillance type of activity, then it may be very hard to link episodes between one episode and another to even ask the question. Is
Yes.
that along the same lines?
Yes, sorry, I should clarify that. So yes, those tools will be useful when you have a follow-up of a patient. So in a clinical trial, or in therapeutic efficacy survey, but where you're following the patient over time. If you're using this for very routine surveillance, similar to the GenRe-Mekong type framework, where you don't know when that same patient comes back in, maybe they even have a different clinical record number when they come back in, and you can't connect them, that will be much more difficult. Yes.
So actually, can I turn the question to you? Do you think it is feasible in the systems that you have in Cambodia or something to actually be able to connect samples between patients?
Yeah, if there is no choice, because currently like PF, we have another choice to see easily, but differentiate between the recurrent and so on, and reinfection. But for vivax, while currently there is no choice or to differentiate that, we have to thinking about it.
Yeah, it's definitely easier. In the examples I've shown, that's an active follow-up. But yeah, if it was passive, that becomes a different story. And maybe it is possible in lower endemic settings. But yes, as Oliver says, like I think that's something for the NMCPs to inform on.
But to be perfectly fair, I mean, this is only one specific use case. And Sarah also presented other use cases like looking at the variability, like the complexity of infection and so on, which you don't need to be able to tie up the.
Actually, it's good because we can assume on that because nothing perfect because even different time frame, different episode, but we can assume that it is everything is perfect, like complete everything. So we can assume, but at least we know something about it.
Yes, what we really hope is that we'll finally be able to get accurate phenotypes for each patient. And maybe now we can finally identify chloroquine resistance markers or other resistance markers. Whereas at the moment, when we do association studies, they're very messy and we never know if our phenotype's clean enough.
Sorry, time.
Maybe skip your question, but sometimes for me, difficult to differentiate between regression and relapse. So by genetic analysis, you can distinguish.
Yeah.
Yes, that's the plan, definitely.
Yeah. Okay.
And modeling of the time to event.
So when I look in our data, we follow the PV patients. So sometime after 1 month, the same patient appear parasite. Yeah. So we think relapse, but sometimes we don't know. Maybe it's recurrent. So I really curious to know if exactly it's relapse or regression. Then we can based on to know
Yes,
that.
definitely. If you have that information and when Dr. Nian does the typing, it'll be wonderful to have a look at that data and see how we can optimize it for to answer your questions there.
Customers. Yes, Dr. Kim.
Yeah, thank you so much for presentation. Really nice for us. And I would like to know about day zero for the vivax, right? You show that day zero is like a relapse, right? Relapse and like this song.
Should I bring a slide back?
Yeah, okay. Can because why I would like to share this. Right now we collect the sample for vivax day zero day 28 and day 90. Yeah, but for day 0 show relapse first, right? Or resistance. And then another day, when the patient come back, they have the parasite. We want to know the new infection or reinfection or liquidistant, something like that, right? But yes,
So, but you cannot say on day 0 is going to be so day 0. Then you compare
another day.
later episode. Then you can tell it's a relapse or recrudescence.
Yeah, we would, for day 0, it's very true. That could itself be a reactivated liver stage, absolutely. But without the preceding infection, it would be difficult for us to tell. So it really requires that pairing at the moment.
Yeah.
I think you might need, you might be wanting other, maybe even serological type assays if you really wanted to know about that day 0.
So this one, you always need to compare two infections.
Yeah.
The first time you cannot know.
Yeah,
But
yeah.
later on, you can say, is it likely to be the same mosquito that infected these two?
Mm.
Yeah, I think that's the question you can answer. Then you will know maybe second one is a relapse.
Yeah, I think so. Yeah, thank you so much.
Excellent. Okay, there's a question from online. Is there work done on serological method to differentiate relapses or from infections like antibodies to sporozoite surface antigens? And if so, how does it compare to using haplotypes? And could this in some way complement the methods?
Yeah, that's a lovely question. Actually, yeah, just now when I mentioned serology, that's what I was thinking of. Work by Rhea Longley and Ivo Mueller that has actually they've developed serological tools that can give you an idea of whether or not you've had a liver stage infection. And I think they could very well be complementary. We haven't brought the two together as yet, but yeah, that's something very worthwhile looking into.
Any other questions from the audience? Okay, well, thank you very much, Sarah, for your impressive work.